Mendel's study of inheritance was based on careful observations of pea plants, but pieces of the heredity puzzle were still missing. Hi, and welcome back. In today's biology segment, I'm going to talk about meiosis.
Meiosis. Genes, chromosomes, and numbers. Organisms have tens of thousands of genes that determine individual traits. Genes do not exist free in the nucleus of a cell. They are lined up on chromosomes. Typically, a chromosome can contain 1,000 or more genes along its length. Diploid and haploid cells. In the body cells of animals and most plants, chromosomes occur in pairs. One chromosome in each pair came from the male parent, and the other came from the female parent. A cell with two of each kind of chromosome is called a diploid cell, and is said to contain the diploid or 2n number of chromosomes. Organisms produce gametes that contain one of each kind of chromosomes. A cell containing one of each kind of chromosome is called a haploid cell and is said to contain a haploid or n number of chromosomes. Why meiosis? When cells divide by a process called mitosis, the new cells have exactly the same number and kind of chromosomes as the original cells. Clearly, there must be another form of cell division that allows offspring to have the same number of chromosomes as their parents. This kind of cell division, which produces gametes containing half the number of chromosomes as a parent's body cell, is called meiosis. It occurs in the specialized body cells of each parent that produce gametes. Meiosis consists of two separate divisions, or we can say that it has two phases, known as meiosis 1 and meiosis 2. Meiosis 1 begins with one diploid 2n cell. By the end of meiosis, there are 4 haploid cells. These haploid cells are called sex cells or gametes. Male gametes are called sperm. Female gametes are called eggs. When a sperm fertilizes an egg, the resulting zygote once again has the diploid number of chromosomes. The zygote then develops by mitosis into a multicellular organism. This pattern of reproduction involving the production and subsequent fusion of haploid sex cells is called sexual reproduction. The phases of meiosis. Meiosis 1. Interphase. The cell replicates its chromosomes. After replication, each chromosome consists of two identical sister chromatids, held together by a centromere. Prophase 1. The DNA of the chromosomes coils up and spindle forms. As the DNA coils, homologous chromosomes line up with each other, gene by gene along their length, to form a four-part structure called a tetrad. A tetrad consists of two homologous chromosomes, each made up of two sister chromatids. The chromatids in a tetrad pair tightly. In fact, they pair so tightly that non-sister chromatids from homologous chromosomes can actually break and exchange genetic material in a process known as crossing over. Crossing over can occur at any location on a chromosome, and it can occur at several locations at the same time. Metaphase 1. During metaphase 1, the centromere of each chromosome becomes attached to a spindle fiber. The spindle fibers pull the tetrads into the middle or equator of the spindle. This is an important step unique to meiosis. Anaphase 1 begins as a homologous chromosome, each with its two chromatids, separate and move to opposite ends of the cell. This separation occurs because the centromeres holding the sister chromatids together do not split as they do during anaphase in mitosis. This critical step ensures that each new cell will receive only one chromosome from each homologous pair. Telophase 1. The spindle is broken down, the chromosomes uncoil, and the cytoplasm divides to yield two new cells. Each cell has half the genetic information of the original cell because it has only one chromosome from each homologous pair. However, another cell division is needed because each chromosome is still double. Meiosis 2. Prophase 2. A spindle forms in each of the two new cells and the spindle fibers attach to the chromosomes. Metaphase 2. The chromosomes still made up of sister chromatids are pulled to the center of the cell and line up randomly at the equator during metaphase 2. Anaphase 2 begins as the centromere of each chromosome splits, allowing the sister chromatids to separate and move to opposite poles. Telophase 2. Nuclei reform, the spindles break down, and cytoplasm divides. And here is an overall illustration of the meiosis process, where you have meiosis 1 and meiosis 2. And these are the illustrations, and on the back you can see photographs of how it looks in real life. Non-disjunction. Although the events of meiosis usually proceed accurately, sometimes chromosomes fail to separate correctly. The failure of homologous chromosomes to separate properly during meiosis is called non-disjunction. In one form of non-disjunction, two kinds of gametes result. One has an extra chromosome, and the other is missing a chromosome. The effects of non-disjunction are often seen after gametes fuse. In humans, if a gamete with an extra chromosome number 21 is fertilized by a normal gamete, the resultant zygote has 47 chromosomes instead of 46. This zygote will develop into a baby with Down syndrome. Polyploidy. Organisms with more than the usual number of chromosome sets are called polyploids. Polyploidy is rare in animals and always causes death of the zygote. However, polyploidy frequently occurs in plants. Often, the flowers and fruits of these plants are larger than normal, and the plants are healthier. Many polyploid plants, such as the sterilite banana plant, are of great commercial value.
I hope you found this video useful. Thanks for watching, and until the next one, bye!